each organism has kind of a main way of how its cells become or go through the specification process. So really this is all this is just to help you understand some of the mechanisms of development. So these are the three kind of main concepts. So again, autonomous specification. In organisms that undergo autonomous specification, they have the ability early on in their cells to already know what they're going to become. So if you separate them from the other cells, they'll still become that cell type or group of cells. And the reason for that is because the RNA that's in the cytoplasm has been sequestered to various areas of that cytoplasm so that when the cell starts undergoing mitosis and cleavage and generates the cells, as the cytoplasm starts partitioning itself off, each of the cells that result from that partitioning are going to have their own proteins or morphogenic determinants. As we talked about morphogen gradients before, they're going to have their own proteins that will tell the cell what genes to turn on. So it doesn't necessarily need interactions with other cells to become what they're going to become. We'll see today how that's not always the case, especially when dealing with vertebrates, which is where most of the development that we're going to be learning about, barring one or two examples, is going to be at. Okay? So whether it's a sea urchin or whatnot, these cells, when you remove them from their native environment, will still continue down the path that they were meant to go, even without interaction with other cells. That's autonomous specification. Why? That's due to the internal cytoplasmic components. What am I talking about? I'm talking about the mRNA and the subsequent proteins that get translated from that mRNA that was originally set up in the oocyte or the egg. Okay? Again, morphogen gradients. A morphogen is a protein that has the capacity to cause a change in the cell or cells that it comes in contact with. We're going to see a lot of different morphogens or signal transduction pathways that are signaled by morphogen gradients today. So again, a morphogen gradient, because of the natural process of diffusion, the cells that are secreting or making the protein will have the highest concentration of that morphogen. The cells adjacent to those that are affected by this morphogen gradient there's certain levels of this that are going to cause certain genes to turn on. As you start getting further and further away, there's a threshold, and that's the key here. There's a threshold at which point the cells at a lower amount either won't respond to it all or respond slightly differently than those adjacent to it. So just the mere distance, and this is again where gastrulation or morphogenesis comes into play, is that as cells move around, the distance or the location in which they are in respect to one another can and does matter. And then ultimately you have cells that have little to no effect on the morphogen gradients, but even low levels of these things are important when combined with other proteins or other combinations of other morphogen gradients, as we'll show. Okay. Now, syncytial specification. Again, this is one that is pretty much in insects. And one of the prime examples that we use for this is Drosophila. And really the main difference between syncytial specification and autonomous specification is that you still have these cytoplasmic determinants, these morphogens, or the proteins that are made from these RNA that are sequestered within the cytoplasm. The difference is the cell will actually go through multiple stages of mitosis, but not cytokinesis, and generate a number of these nuclei before cytokinesis even occurs. What's happening is these morphogenic gradients are being set up so that when the cell starts partitioning off into smaller cells, the gradients that have been set up over a period of time then become encapsulated within the individual cells, and that creates patterns of development that will play a huge role later on. In fact, we show that if you disrupt some of these morphogen gradients, certain parts of the Drosophila won't even form if you disrupt the flow of these morphogens going from one side to the other and then being, restricted to the cytoplasm of the individual cells. Okay, so more on that later when we get into some of the details of Drosophila. Now, you and I, as well as pretty much all vertebrates, for the most part are under what we call conditional specification, which is the fact that the cell's location and interaction with one another does matter. So in the initial stages, we don't have things that are sequestered to certain parts of our cytoplasm that ultimately tell the cell what to become. In fact, in the first few stages, even that inner cell mass has the potential to still become every cell in an organism, which is why when you have monozygotic twins, the inner cell mass of the developing embryo, when it splits, each one can become its own embryo. So early on, the cells are not specified at all. It all matters on what type of interactions they end up having later in reference to the placenta, their implantation of the uterus. Again, I'm talking about in our case. In other cases, they're a little different. We'll go through some of those later on.